I thought what I would do is tell you about 4 stories that we're developing, one of which just came out, the last one which just came out. And that is how well do the preclinical models that we all use cover the molecular profiles found in tumors and the TCGA and the George and related data sets. Show you some work we've been doing on dropout screens to identify which could be key stem cell pathway genes that are required for small cell lung cancer growth. Show you the latest information we have on targeting one of the lysine demethylases, KDM4, as a potential therapeutic target for small cell lung cancer. And then end by showing the results on preclinical studies of a telomere targeting drug, 6-thiodeoxyguanosine, for depleting both cancer stem cells and also potentiating radiation. And interestingly enough, as you will see, that this depends on also having an intact immune system. Well, I thought I would start out first by how I got into this, into genetics and all. And I was a medical student just starting at Stanford, and they had one of those introductory dinners. And it was at a professor's house. And this gentleman sitting across from me at that time, he was wolfing down a plate of spaghetti and firing questions at me right and left. And it was Joshua Letterberg. And he said, Minna, what did you do this summer? And I said, well, I did computer programming. Now we're talking punch cards and Fortran. That really dates me. And he said, Minna, that's great. Monday, you're coming to work in the Department of Genetics. And what's obviously he won the Nobel Prize in 1958 for discovering bacterial conjugation. He was the founding chair of genetics at Stanford. But what a lot of people don't know is that he was the real power behind all of computational biology and artificial intelligence at Stanford. This is way ahead of the time. Obviously, Stanford is well known for this now, but he was the one that said, if we're going to do this in biology for any other things, we need to have computational power. And he foresaw a lot of this stuff way ahead of time. And he really taught me about attacking deep and important questions. And most importantly, gave me a job in the Department of Genetics at Stanford from my first day as a Stanford medical student. And that happened to be in the lab of Lenin Lee Herzenberg, who shortly after I left developed the fax machine, and he won the Kyoto Prize. He probably should have won the Nobel Prize from that. And I spent 5 great years in that lab learning about genetics and actually mouse genetics and immunology. And what was so great was the way Lin and Lee taught us to critically think about experiments. And even more important, there were all kinds of famous people in immunology coming in and out of this lab. And for Lin and Lee, it didn't matter whether you're a professor or a medical student. It was really what about your ability and your thoughts and not your position that they considered important. And they really had a very nurturing, fun, and interactive lab environment. Highly critical about your work you presented, but if you did good stuff, they didn't care who you were. So the first topic I'm going to tell you about is the current work we're doing to see how the preclinical models, cell lines, and patient-derived xenografts cover the molecular data sets. And this, of course, makes me think about my long-term colleague, my older brother, Adi Gazdar, that worked with for decades, distinguished professor of pathology here at UT South, NCI and UT Southwestern. And we worked together for many years before he passed away. And of course, he was foundational in developing all of the early tumor cell lines and xenografts and their characterization. So clearly, preclinical models are needed. We all use them for experimental studies and to be able to provide cell lines and xenografts to the general community to allow others both to stimulate research and allow others to validate findings. And when one looks at the general nature of histology, mutations, or expression remains between cell lines and xenografts and the patient tumor samples, there's usually excellent, but it's not perfect concordance. It's probably for the main features is probably 80 or 90%. And also, if you look at mutations and messenger RNA characteristics of these over time, they're reasonably stable, particularly for the key drivers, which are almost 100% stable. But to be most useful, we need ways to match these to the large deposited tumor data sets that are also clinically and molecularly characterized. Now, the tumors in TCJ and George data sets are not those that went to create these lines. But the question is, if you're working with a line, which of these other tumors do they most closely represent molecularly? And so we're doing this by comparing the mutations that are found in 700 cancer-relevant genes and also RNA-seq expression analysis for 2,000 of the most variably expressed genes. And so we try to do a matchup, which preclinical model is matched to which individual tumor models in these other data sets. And this has been a longstanding collaboration with Jack Roth in his lab, and then for small cell, Ben Drabkin, and led by Luc Girard, one of our faculty members. And for the small cell lung cancers at UT Southwestern, this has been Ben and his postdoctoral fellow, Shiroshi, and then Victor Stasny, who's a laboratory tech par excellence, who worked with Dr. Gazdar for many years and now with us in creating these models. And of course, at MD Anderson, this has been a partnership with our SPORE as well, with Lauren Byers and Carl Day, who've developed a lot of new small cell patient-derived models as well. And so we can do this by, I won't go into the details, comparison by expression of these variable expressed genes, how well are they coordinated? And then you could look at mutations and you can get a concordance in for that, and then compare both the expression and mutations. And here's a heat map showing for 1,000 or more TCGA specimens and all the specimens in the two deposited George data sets for both large cell neuroendocrine cancers, 3 different data sets for large cell. Then the first other George data set that was on resected tumors, and then the more recently published one on metastatic tumors. Those are all on the columns. And then if you look at the rows there, we've color-coded them for non-small cell lung cancer cell lines, for adenosquamous, or otherwise just the grays are for non-small cell, small cell lung cancer lines, non-small cell lung cancer PDXs, and small cell lung cancer PDXs. So if, I don't know if you can see my mouse there, but if you take adenocarcinoma lines and you match them against TCGA, and the black means that they would be highly concordant, white would be less, you see this panel of concordance. And then interestingly enough, there's some concordance with squamous, but then if you come down to the squamous, you'll see that more, and there's less with small cell. And then we'll just go to the small cell lines and small cell PDXs. As you see, there's very high concordance with the George tumor data sets. And another way to, but as you can see, there are many where they're not perfect concordance, even though they have scored. And so here in a plot of density versus this concordance, you can look at for adenocarcinoma, blue, squamous in red, the 2 George data sets in yellow, the George large cell neuroendocrine. So there are the three George data sets there. And so approximately 70%, so in these concordance here of 0.6 and 0.7 would be pretty highly concordant. In the non-small cells, about 70% of the tumors in the TCGA data set, be they adeno or squamous, have a very, a very good molecular match, but about 30% don't. Now, I'm not going to show you this, so that, and this is from many different labs trying for both PDXs and cell lines. So there's at least 30% of non-small cell lung cancers that we know develop clinically evident tumors that for one reason or another have not been able to form a PDX or a cell line. And it turns out, this may be slightly higher for adenocarcinomas than squamous. But when we look at the molecular characteristics of those, it turns out the expression profiles of the tumors that we are not able to derive these preclinical models from have much better prognosis than the tumors which we are. That may or may not be surprising, but it clearly shows you if we could understand what this difference was, it would be a potential new therapeutic. Now for small cell, there's less. As you can see, we probably have 90% of the samples have a match, 10% yet don't. And you can see that's also true for the large cell neuroendocrine. So this is kind of the state of the art. And so in designing your trials, you can go into these data sets. And now, obviously, we have over 100 small cell models, probably 150 when you count all the cell lines and the PDXs. But the question is, you could then pick out subsets of those to work on to both help your grants and focus your research. And this just shows some of the concordance. First with the limited stage for the small cells with that. And as you see from these other multiple color coded, there are some, for example, squamous PDXs, which gets mixed up or have some match with some of the small cells. And likewise, a couple of the adenos with the small cells when we're looking at the cell lines. And that's the same thing for the data set too. So this is just summarizing all of this. We've done all this. And I think these are going to be very useful. We're getting all the websites set up so people can go in and have this. And we want to expand this to PDXs done from other sources as well, besides the UT Southwestern and MD Anderson data sets. And again, well, I didn't show you this, Ling Shai at some time in the future can give you, she's done a similar thing for the molecular annotation of GEM models, and she now has close to 800 different molecularly annotated GEM specimens derived from multiple different labs to put in a similar type of data set. Well, I thought I'd pause for a second and talk to you. This is a radiation oncologist, Eli Gladstein, again, another person that's been like my older brother who passed away in 2020. And you will not necessarily know that, but radiation oncologists and small cell lung cancer clinical researchers will remember that he was one of the pivotal figures in small cell lung cancer clinical trials looking at combined modality chemotherapy and radiotherapy. And on the right is a picture of him and two other very famous by the way, Eli trained over 20 chairs of radiation oncology throughout the United States. On Eli's right is Norm Coleman, who fantastically important figure in radiation oncology, headed up all of the NCI's response to the Fukushima disaster, as well as others, and was a triathlete and unfortunately died after developing a very aggressive sarcoma a few months after he just participated in an Ironman event. And on Eli's left is Alan Lichter, former president of ASCO and a very famous radiation oncologist who was with us at the NCI and participated in all our first clinical small cell lung cancer combined modality therapy trials. So I want to tell you next about trying to use in vivo dropout screens to determine the role of potential key cancer stem cell targeting pathway genes for that could be potential therapeutic targets for small cell lung cancer. And this work is led by two very important scientists in our group, Longshan Li and Kenneth Huffman, as shown here. Well, people have known, and several different centers have known for a while, that small cell lung cancers are very enriched with a subpopulation, maybe 10 to 50% of the tumor cells with a stem cell phenotype referred to as cancer stem cells. Now, in contrast to non-small cell lung cancers, where in many cases they're 0.1, 0.5, maybe 2 or 3% of the total cells in the population may have these phenotypes. And these appear to be the cells that form tumors and metastasize. So the ability of a cancer stem cell and small cell to form a tumor in a xenograft is maybe 500 or 1,000-fold enriched compared to other cells within that same tumor cells within that same tumor that don't have a cancer stem cell phenotype. And there are several different genes and many potential targets that help determine this cancer stem cell phenotype. We need to test small cells from many different patients to see how they would respond to this targeting. And of course, what is really important is not what happens in vitro, but what would happen in vivo in a tumor. And so we did this with generating a lentiviral library that targeted 42 different stem cell pathway genes. We put these into 8 different small cell lung cancers. We then formed xenografts, and we did parallel studies, both with the cells grown in vivo and then in parallel grown in vitro. And of course, this also gives us the ability to test many potential therapeutic targets at the same time. Now, some of the technical details I want to go into that are important for planning these experiments. You may say, why didn't we do genome-wide? Well, it turned out that we wanted to have many different targets for each gene, each with a separate barcode, to allow them to be identified by sequencing. We wanted to do this with a multiplicity of infection of about 0.3. This is from Poisson distribution, so that on average, a tumor cell would get none or only one of the short hairpins. So any one tumor cell would only be testing the impact of one of these barcoded genes. We wanted about a 2,000-fold representation. That would mean we'd have to put enough tumors, we're talking about 10 to the 7th or more tumor cells in, so that any one short hairpin would be tested in about 2,000 tumor cells, which we knew would be enough to give us a tumor. And we wanted to do this in many replicates on many different types. We wanted to see in vitro versus in vivo. And for each experiment, we had 5 mice for each tumor, so we had 120 different assays. And then the tumor DNAs would be then looked at initially to see what was there to begin with, and then at the end, down the line, to see which dropped out, which would then be essential for their growth. And it was also critically important that if we're going to score a gene as a dropout, that we had to have multiple independent short hairpins targeting the same gene that had dropped out. Now, I won't go in, I just put this here, and I'm happy to share. These are all well-known genes that have been involved in cancer stem cell work. In addition, we put in the lineage transcription factor, ASA1. We now wish we'd put in all of the others as well. And then we put in positive toxic controls. So there were very large numbers of samples that went into this experiment. And in addition, we also wanted to know, is it possible that any of the tumors, because small cell lung cancers can have a lot of mutations, would any of these genes have amino acid sequence altering mutations? And a few did, and they're shown here. We don't think that had any impact, but we have the ability, in case there was something weird about a stem cell gene having a mutation in a particular tumor, we could test that. And just to give you a flavor, in one of the things for both in vivo and in vitro dropout, this is a way of seeing how much dropout there was. And in the in vitro one at the top, these are average of 3 replicates and initially, so you know which barcodes actually got in and could be detected. And then at passage 9, there were 3 replicates. So they to see the dropped out. And when we have a dropout of down at -4 or -6, this is really dramatic dropout. And you can see here's another volcano plot to show this on the right. And then in the in vivo dropout, there were initially 3 replicates from a tumor before when the tumor was initially started, and then 15 replicates later on when it was harvested. And here are the genes that dropped out in vitro and vivo. I won't go through it. You'll see another comparison here, but some are similar, but there are quite a few that can be different. Again, since these were stem cells, the null hypothesis was that every one of these was going to be important and they would all drop out. Well, here's a heat map summary of showing these genes and the different lines and whether they expressed ASCL1 and NeuroD1. You can see we had, for this particular one, we had a spectrum. And the red means that there was highly significant dropout. So at this top, H2O81 and ASCL1 high expressing, that expressed some NeuroD1, by and large dropped out just about every one of the stem cell genes. And then it progresses all the way down to SHIP77, which has always kind of been an outlier, but also H60, which has been widely used. And you can see that in many cases, these stem cell genes did not drop out. And in fact, we knew that a key markers of cancer stem cell phenotype were ALDH 1A1 and 1A3. And so we were very interested to know whether these are enzymes that could be potentially therapeutically targeted. And you see in some they are required, but in others they're not. And similarly for promenin, another well-known gene. Interestingly enough, ASCL1, which had been a major lineage oncogene target, there were clearly some which in vivo was highly required, but in others that it was not. Now, it might not be surprising if this was a NeuroD1 driven tumor, as I'm showing you. I should probably have had more errors, but there are others where it was clearly highly expressed, but still not required. And here's just another, I haven't gone case by case for in vitro, in vivo comparison of all of these, but the point of fact, in some cases like 2081, a lot of matches, but there were several where they were required in vivo, but not, interestingly enough, in vivo. And you can see that there are some discrepancies, and we've gone through to look at these. So there clearly can be quite different growth requirements from the in vivo versus in vitro situation. And of course, this is important because if you go into DepMap, those are all essentially in vitro dropout screens for in their CRISPR and short hairpin screening. So we've been able to successfully do this in 8 different cases. There were differences between in vitro and vivo, and there were also intertumor differences in which cancer stem cell genes were important for a particular small cell lung cancer. Well, I want to pause here for another important part. Henry Kaplan, who was one of the major founding figures of all radiation oncology, developed the linear accelerator at Stanford. He developed some of the first treatment for Hodgkin's disease and the whole methodology for doing clinical trials. He was a very renowned radiation biologist and tumor virologist, and he had established meticulous conduct of clinical trials of integrating the laboratory and the clinic. I did 6 months with radiation oncology when I was a medical student. And during that time, I personally worked up 105, count them, 105 new patients with Hodgkin's disease. Now, many medical oncologists or hematologists in their career may not see that number, but the number of patients that were flowing into Stanford for that were amazing. He was a very important mentor, took a group of us under his wing and got me my later jobs. I subsequently learned that the main job I got at NCI from Vince DeVita was Vince called up Henry, and he because of Vince's interest in Hodgkin's, said, Henry, I've got this branch that's dealing with lung cancer. I'm going to close it down. He said, is there anybody you know that could do it? He said, well, there's this young guy that's there at the NIH, John Minna, and why don't you hire him? I only learned that 20 years later. So at age 33 or 34, I was given a battlefield promotion, equivalent of being promoted up to a colonel or a Navy captain to run this NCI program. And then the other one was Saul Rosenberg, a medical oncologist, one of the founders of medical oncology. And again, another person that took me under his wing, got me on the career to going into medical oncology, was a very important, he and Henry Kaplan ran these Hodgkin's trials. Henry was more on the trials part, and Saul was more on how you deal with individual patients and the importance of studying and knowing the individual patient, but also important about knowing my own strengths and weaknesses. Subject for another day, but he was extremely influential and a revered figure in medical oncology. Well, I want to tell you now about our work trying to identify new therapeutic targets in small cell lung cancer that would have different response phenotypes than those to etoposide, and that would also work in etoposide resistant small cell lung cancer. And this is work led by Elizabeth Martinez, who's Associate Professor of Pharmacology here at UT Southwestern and in the Hammond Center, and a PhD student that we co-mentored, Aidan Noonian. Now, this is kind of the bottom line of all of this, is this paper, it's wending its way through to get accepted. So some Jumanji lysine demethylase KDM family members, such as KDM4, are highly expressed in small cell lung cancer. There are 30 or so different family members of this. These are known to be help regulate the epigenome by performing their lysine demethylase inhibitors. These would be erasers. And it's been known that we found out that these inhibitors of some of these enzymes will block the malignant growth of small cell lung cancer. We found that etoposide resistant small cell lung cancers, as you know, this is a major problem. Initially, small cell lung cancers and patients respond to platinum etoposide, but then within a year become almost universally resistant. So we found that these very resistant small cell lung cancers were very sensitive to this Jumanji inhibition. We also found that these inhibitors decreased the expression in small cell lung cancers of key neuroendocrine markers like INSIM1, segregagranin 3, but also lineage transcription factors like ASC1 and NERD1. I'll show you some of that. And if you specifically knocked down or knocked out KDM4, it recapitulated the finding of these inhibitors, because the inhibitors are not necessarily absolutely isotype specific. And I'm not going to go into this, but subsequently Liz found that the small molecule mediated inhibition of KDM activated ER stress genes and ER stress signaling and triggered apoptotic cell death. And importantly, small molecules inhibitors, including some of those like TAC101, they're into or transiting into the clinic, could block small cell lung cancer xenograft growth. So here's a heat map showing for a large number of small cell lung cancers over here, and then a whole cadre of immortalized lung epithelial cells, normal lung epithelial cells with and without potentially some other changes. Here are all of the different KDM family members that we looked at, and uh, uh, KDM 4A will be this one right here will be the ones we're talking about. The structure is shown here. I'm not going to show you the results, but there's another one of these KDM 5B. I don't know if I can find it on here, which we also knocked out just as a control and had absolutely no effect. So we could see high expression in many, but not necessarily all, small cell lung cancers. And here's the structures of these different drugs. GIB04 was developed and designed by Liz. It's a pan-Jumanji histone demethylase inhibitor. And then GSK drug ST70, which is a KDM4 family inhibitor. This one is a KDM6 family inhibitor, and the TAC101 is a pay. There's several different KDM4 family members, and it's a pan inhibitor. Well, this is just to show you if you take a panel of these lines and is looking at the spectrum of response of small cell lung cancers to this. And it's not necessarily comparing one drug to the other, but a resistant tumor to a sensitive tumor for these from here over to here or from here over to here. And as you can see, in all cases, there's about 100-fold difference between a small cell lung cancer that's sensitive and resistant. And for example, this is GIB04, which is the most sensitive one here, but here is TAC101, which would be way over here as well. But again, from sensitive to resistant, there's about 100-fold difference. And then shown here in 2 small cell lung cancers is with an IC50 dose that you could actually see that treating with these inhibitors actually inhibits the enzyme activity measured in relative K3, H3K9 methyl 3 demethylase activity for both of these, so that they do provide on-target inhibition of this enzyme activity. Well, here's what I consider some of the key data. So if you look at the large data set developed by NCI, you saw sensitive small cell lung cancers that were sensitive and resistant to etoposide. And then there were other drugs in that large 700 drug panel screen that had sensitivity and resistance. But in all cases, there was a highly correlated, high correlation between etoposide sensitization, sensitivity and response phenotype and resistance response phenotype. So you were basically seeing a drug sensitive or resistant phenotype determined by etoposide, but then mirrored by everything else. But in these comparisons here, correlation coefficients, or these heat map comparisons here, you can see these Jumanji inhibitors had no correlation with etoposide sensitivity and resistance. And here's a panel of small cells that are very sensitive to etoposide. Some are sensitive and some are resistant to these Jumanji inhibitors. And there's a whole panel down here of resistant small cell lung cancers that are resistant to etoposide. And many of these are now sensitive to these Jumanji inhibitors. So if one now wanted to either use this as combined therapy at first or at the time of relapse, these inhibitors would have the potential to do that. And so besides doing the drug targeting, we looked at CRISPR knockout of the genes. And interestingly enough, we found in the case of this was a NeuroD1 high one, that if you knocked out KDM4, that you also could see a decrease in these neuroendocrine genes. And likewise for NeuroD1. And if you targeted these with drugs in an ASCL1 high one, you would see depletion. So one possible mechanism could be the epigenetic moderation using these epigenetic targeting drugs of the lineage transcription factors as well. And this was also then seen in xenograft studies. Here's a xenograft knockout of KDM4, which shows impressive inhibition of xenograft growth, and then treatment of the same things with these various drugs showing this inhibition. And then also looking in vivo at the loss of expression of both the neuroendocrine drugs or the lineage transcription factor in an ASCL1 high tumor with treating of these. Well, the final mentor that I want to tell you about is somebody that I spent 7 years with at the NIH, and which was Marshall Nirenberg, who won the Nobel Prize in physiology or medicine, obviously for cracking the genetic code. He did this with Karana and Holly, and how it functions in protein synthesis. And Marshall taught me how to crystallize a project, be fearless, to collaborate, and there will always be new things to do. And I'll just tell you a story when I walked into his office and said, "Hi, Mark." By the way, Henry Kaplan was the one that wrote and got me my job with Marshall. And I said, "Marshall, I'm John Minna, and so I just finished my internship and residency and want to work on this." And I say, what should I work on?" He said, "John, you've come at the perfect time. Mal Martin has just discovered that SV40 virus can immortalize cells. And even better than that, Phil Nelson's in neurobiology. He just discovered that if you put a needle into cells, you can get you can see firing in neurons. And if you have two neurons connected together, you can detect a synapse." So he said, "John," he said, "order up a bunch of pregnant mice, take the fetuses out, take their isolate their brains, put SV40 virus on them, you'll immortalize all the neurons there, and then clone all of those neurons that are now growing, clone them, and take clone A and put it with clone B, and take that in a plate down, let it grow together, and down to Phil, and have him look, and then put A with C. And Phil's going to show that A is going to form a synapse with B, but not with C. And John, you'll figure out the synaptic code." It was literally in that space of time that he told me that. So I said, well, he won the Nobel Prize for doing this for the genetic code. Why not figure out how the brain works? So I walk out across the hall into a room that was probably about the size of the office I'm in now, in which were 10 postdoctoral fellows, 2 of which would win the Nobel Prize, all of which would become chairman of biochemistry or molecular biology. They said, "John, what did Marshall tell you to do?" And I told them. There was this pause, and then there was laughter, and people were literally rolling on the floor. They said, "John, Minna, you don't have any idea. This is just about impossible to do." Well, we tried that. That didn't work, but I went on to make the first somatic cell hybrids between neuroblastoma cells and normal neurons, which did work at the NIH. And so, Ben, well, I have something more. I'm going to just take a stop here and take questions. And this paper that Ezra Akbay and Jerry Shave has just come out in Nature Communication that talks about using Jerry's drug, 6-thiol-DG. It's not 6-thioguanine. It gets selectively incorporated by cells that express telomerase, which small cell lung cancers have a lot of, and it targets the cancer stem cells in them, and also gives you an amazing effect to radiotherapy. But interestingly enough, this requires an intact immune system. So I'll stop there and can take any questions